So let's talk about how the PDH complex is regulated, how the rate of this reaction is controlled. So basically, it is pretty common that the rate of reaction of any particular enzyme is controlled through covalent modifications, through the addition or removal of a phosphate group. So it is this for this particular reaction, we're going to be talking about E1. So instead of just like covalent modifications on E2 or E3, this reaction is regulated or controlled by modifying the enzyme E1, which, to remind you, is pyruvate decarboxylase. So there's two further enzymes that are going to either phosphorylate E1 or dephosphorylate it, thus activate or deactivate it. And this is how it goes. So the PDH kinase is going to phosphorylate and there's a typo here, but it's basically going to phosphorylate and thus inactivate E1, whereas the PDH phosphatase dephosphorylates and hence activates E1. So the kinase adds a phosphate group, as you can see right here, phosphate group. So the kinase kind of adds a phosphate. Kinases in general just add phosphates. And the phosphatase dephosphorylates it, and when the phosphate group isn't attached, that's when it's active. So the kinase, you can see in this diagram that the kinase is activated by high energy compounds, such as ATP, such as acetyl-CoA and NADH. There is an abundance of these compounds in the cell when the cell has plenty of energy, when it is in a state of high energy. And when you already have so much energy, you don't really want to produce more. So these high energy compounds activate the kinase, which is going to phosphorylate the PDH complex and thus inactivate it. So the PDH complex won't produce acetyl-CoA, which won't go into the Krebs cycle, and so more energy won't be produced because the cell already has plenty. Other than that, the pyruvate, as you can see in the diagram right here, it kind of inhibits the PDH kinase. Why? Because when you have an abundance of pyruvate, you just want to get rid of it. You want to make sure that most of it is used up in the reaction, and so the PDH kinase is inhibited. The phosphatase would then get active. It would dephosphorylate the PDH, thus activating it, which will use more and more pyruvate as a raw material converted into acetyl-CoA, so the pyruvate doesn't get a chance of accumulating, because pyruvate accumulation can cause problems, can cause neural defects, can cause eventual death, and you basically want to be safe from that. Arsenic poisoning. Now this is an important topic. Arsenic poisoning is mostly due to the inhibition of enzymes that require lipoic acid as a coenzyme. So we just talked about how the PDH complex required 5 different coenzymes, the mnemonic for which was the lovely coenzymes for nerds, and the lovely L, lipoic acid. It requires lipoic acid too, PDH. And so arsenic poisoning is going to affect PDH, because what arsenic basically does is that it binds to lipoic acid, forms pretty strong bonds, and kind of just renders this coenzyme useless. The lipoic acid that is now bound to arsenic is unable to act as a coenzyme for the PDH. So the PDH, now that it's missing a coenzyme, the complex itself will not work anymore. And when it refuses to work, the pyruvate isn't going to get converted. The pyruvate isn't going to get converted to acetyl-CoA, and so the pyruvate is going to accumulate. And once that happens, as I talked about it earlier, accumulation of pyruvate is very dangerous. It causes neurological defects and disturbances and will cause eventual death. Okay, now we're moving on to the Krebs cycle. So you may have noticed the Krebs cycle is called cycle, and I'm going to explain to you why it's called cycle. There's a particular reason for this. A cycle, if you look at the definition of the word cycle, it basically just means a series of events that are repeated in the same order over and over, or a complete set. So as you can see in this diagram, you have acetyl-CoA, which has 2 molecules of carbon. You have oxaloacetate, which has 4 molecules of carbon, so 4C and 2 carbons. And both of these kind of combine together to form citrate, a 6-carbon compound. This citrate goes through an entire cycle. It produces lots of produced carbon dioxide. It will produce NADH. It will produce GTP, FADH, more NADH. And after it has been through the entire cycle, we're going to get oxaloacetate back again after it loses 2 carbons. So a 6-carbon compound getting converted into a 4-carbon compound. And this is exactly why it's called a cycle, because it's regenerated. It is regenerated in each cycle, the oxaloacetate. Okay, so a short overview of the cycle. We're going to talk about the products of this cycle and the raw materials needed. Basically, you literally just need acetyl-CoA. That's about it. And the oxaloacetate, which already came from the citric acid from the cycle before, and the oxaloacetic acid is going to combine with the acetyl-CoA, and it's going to form a 6-carbon citrate molecule. Once you have the citrate, it's going to go through this whole reaction, form lots and lots of different compounds, but it's going to give off lots of small molecules. So it's going to give off reduced electron carriers. It's going to give off carbon dioxide, because a 6-carbon compound getting converted into a 4-carbon compound. You're basically missing 2 carbons, right? And every carbon that you're missing gets converted into a carbon dioxide. 1 here, carbon dioxide, 1 here. That's the second, this is the first. And produces raw ATP, produces reduced electron carriers. And let's talk about this in more detail. Again, the products of the TCA cycle, 2 molecules of carbon dioxide per cycle. So what you need to recognize in this particular slide, what's important here is that we say 1 turn of the cycle. So 1 turn of the cycle, or you can say 1 acetyl-CoA combining with 1 oxaloacetic acid, is going to produce 2 molecules of carbon dioxide, 1 ATP molecule, 3 of NADH, and 1 of FADH2. These are the end products of the TCA cycle. Carbon dioxide is obviously a waste product. The waste product basically just, again, excreted by the lungs through exhalation. ATP will give you direct energy. These reduced electron carriers are going to go to the electron transport chain, get reoxidized, and that's how they produce ATP. Now, this image, I understand, pretty daunting. It's a lot's going on here. But basically, in essence, if you were to look at it, it's basically the same. If you were to shorten this, summarize it, oxaloacetate combining with acetyl-CoA, giving you a citric molecule, which is going to give you lots of different compounds, but eventually just give you another oxaloacetate, so that the cycle can keep on going, can continue. Out of all of these 7 or 8 reactions that are shown here, the 2 of most importance would be the conversion of isocitrate to alpha-ketoglutarate and alpha-ketoglutarate to succinyl-CoA. Why are these 2 reactions of importance? Is because these 2 enzymes are controlling the rate-limiting reactions of tricarboxylic acid cycle. I have a whole slide on that. Let's talk about that in a bit. But first, succinate dehydrogenase is also circled or highlighted, put up in a square, whatever. And it's important because this particular enzyme, succinate dehydrogenase, while the rest of all of the enzymes are present in the mitochondrial matrix. Again, we know what the matrix was. Let me just show it to you one more time. Mitochondria, inner membrane, which is the cristae. This part is what the matrix is. So while all of the regular enzymes are present in the matrix, succinate dehydrogenase is present in this membrane. It's present on the inner mitochondrial membrane. It's not in the matrix. It's stuck on the membrane itself, and that is why this one's also important. So let's talk about this and this in a little bit more detail. The rate-limiting step of the Krebs cycle. So, as it says here, in contrast to most enzyme reactions that are regulated by limiting the rate of just one enzyme, the Krebs cycle is regulated by controlling the rate of multiple enzymes, not just one, like regular reactions, but multiple. Just two main ones, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Now, for these reactions,